Hi, all right, we left off talking about the cell cycle and specifically how it relates to mitosis. So let's pick up where we left off. We drew out a cell cycle and we had something like this. We had our G1, we had S, G2, and M. And one last thing I wanted to stress to you guys before we walk into mitosis is that G1S and G2 together, all three of these phases, this is called interphase. This is where the cell is just doing its job, it's copying DNA and doing all this. Obviously, M phase, we talked about already, is mitosis. So as we walk into mitosis, know that this word interphase is for everything outside of mitosis. And now we're going to get into the nitty-gritty for mitosis. So here we go. Just one last recap from last time. So this will be Roman numeral 3. Let's go straight in to mitosis. All right. With a handful of phases within mitosis. So what I want you to know is that we're going to start with one phase after another. So and with each phase of mitosis, it's not a quick, you have your one somatic cell with your copied chromosomes, and it's diploid with sister chromatids. It's not how it just doesn't go bloom like into two new cells. It takes time. You have to go phase by phase by phase because it's a very important process. If this gets messed up, the new daughter cells that get made aren't going to be functional and they might do damage. So we want to make sure that this whole process happens well. So it's happening very timely and it's concise, it's checkpointed along the way, that to make sure that things are splitting correctly. And so every phase kind of makes sure things are working well. So let's talk about the first phase of mitosis. It's called prophase. And with each phase, I'm going to have a picture of a cell and I'm going to describe what's happening in the cell. So prophase is going to have a little bit more background on it first because we've got to start and set the scene. What's happening with prophase, the cell just got out of G2. So G2 just ended, it's entering M phase, and so this is going to be my big cell. And I have my nucleus here, my nuclear envelope. Remember that it's a double phospholipid bilayer, so this is a pretty legit structure. And for our quest into mitosis, I'm going to create a cell with 2 chromosomes. Remember, you have 46, but I'm not going to draw 46 because it'll get messy. So I'm just going to draw 2. And remember, at this point in the game, the chromosomes are copied. So I'm just going to draw them like this. And I'm drawing them kind of lengthened out and long because at this, they're just getting out of G2. Remember in G2, they started, they were working, they were doing their job. If a cell is doing their job well, it's reading the DNA to know what to do. So if I'm a chromosome, I actually have to loosen the DNA within the chromosome so it can be read. Just like a book. I have to open the book so I can read what's going on. If my book is closed, I can't interpret what's happening. So same thing with DNA. The chromosome has to kind of loosen up a bit. Its typical X shape is a very loose X shaped. So I'm kind of drawing these lengthened out for the moment because they're just getting out of working. And at this point in the game, they're being told, stop working, and like everyone stop working, close your books. That's what's being told to the cell. And so the nucleus is telling the DNA, it's time to start bringing it in. Close your books. What other things that's happening during prophase is that there are these structures in the cell, and we alluded to them last time a little bit, but there's these little barrel-like structures in the cell. And these barrel-like structures are responsible for creating the microtubules. Remember that this little pair of structures right here, this is called a centrosome. A single one is a centriole. So a pair of centrioles is a centrosome. but we would just refer to them as centrosomes because they work in pairs. So during prophase, we need to copy these centrosomes. We need another one there because, and this actually happened like at the tail end of G2. So by the time you're entering into prophase, if this hasn't happened yet, your centrosomes get copied. So you have a copied centrosome. And this will become clear why we need two, because at this point in the game, we're developing these microtubules that are going to start to latch onto the chromosomes at their kinetochore region. Remember, we talked about how you have your sister chromatids, and on the outside parts right here, the microtubules are going to latch on here, and they're going to pull these chromosomes apart. And that's what's happening right now. So slowly but surely, the centrosomes are starting to grow these microtubules out so they can eventually make their way to the chromosome to latch onto them. So these right here are the microtubules. And they're going to create something really cool. Eventually, they're going to create what we call a mitotic spindle. They're essentially going to be a web, a network, if you will, of microtubules that are going to grab onto the chromosomes and pull them apart. But that's coming up soon. So this is prophase. And let's just define what we see here so far. So in prophase, here's the first thing that's happening. That the chromatin inside the chromosomes, the chromatin is starting to condense. It's starting to get tighter and packed more. So the chromatin, we're closing the books. So the chromatin in the chromosomes, which are like kind of one and the same, is beginning to condense. And if you want to think of it, it's getting packaged. It's getting tighter and tighter and tighter. So we can actually do something with this and make it easier to work with. It's if I'm trying to split my DNA equally up into two new cells, it's easier if I bring it all in so I could just go and take them apart. If they're all loosey-goosey all over the place, I kind of got to it's like wrangling cats. You actually want to bring them in nice and tight, so it's good. The second thing is that each chromosome is obviously a sister chromatid. It appears as a sister chromatid by this point, and that's great. And what's happening is the duplicated centrosomes begin to create microtubules. So the duplicated, because they got duplicated, they got copied at the end of G2, maybe the very beginning of prophase, it's around that area. So duplicated chromosomes, I'm not chromosomes, I apologize. Centromeres, forgive me. Centromeres begin to make microtubules. There we go. And what's happening is, as the microtubules begin to lengthen and you're making more and more, the microtubules are getting longer, the centrosomes, the pair of centrioles, they're starting to migrate to opposite poles of the cell. So the centrosomes begin to migrate to opposite poles. Of the cell. Like the microtubules are actually, as they lengthen out, they kind of like push them back into different poles of the cell. It's kind of neat. So that's prophase. All right, our next phase is pro-metaphase. So very close to prophase, and oftentimes in a lot of textbooks you'll see prophase and this next phase, pro-metaphase, all tied up into prophase. That's perfectly fine. I'm giving you the honors version of it. I'm giving you a little bit more detail on how it actually happens. It's okay to lump it all up into prophase, but this is what actually, if you want the honors, a little bit more detailed version of what's going on here. So either way is perfectly fine. So for prometaphase, you're starting to get the cell split. So prometaphase. So for prometaphase, this is where it gets kind of fun. Here's my cell. Now my nuclear envelope that was nice and solid in prophase, in order for me to break apart these chromosomes and put them into new cells, I actually have to break down the nuclear envelope for a moment in time. This is highly dangerous. This is the first time the DNA has been exposed to the rest of the cytoplasm in the cell since it got split before. So this is why we shut down everything in the cell as well. We don't want anything interacting with the DNA or working with it or anything. So we start to break down the nuclear envelope, and now the chromosomes are exposed to the cytoplasm. So here's what's going on. You get this breakdown of the nuclear envelope. So you get the nuclear envelope breakdown. And inside, actually, I'm going to draw them a little bit more condensed now, because by this point, during prophase, they were starting to condense, and now you're getting these, you know, they were long, and now they're shrunk in. So we closed all the books, which allowed it to shrink in a bit more. So now, at this point in the game, we have our centrosomes as well. They have completely migrated to the poles by this point. And what's happening, they're growing all these microtubules. Now that the nuclear envelope is beginning to break down, and it's eventually going to go away for a moment in time, but during prometaphase, you're seeing it just start to break down and disintegrate. Now these microtubules that are growing out of the centrosomes are able to go into this nuclear region that once was, and they're going to start latching on to the kinetochore region on the chromosome. So you're going to start seeing this latching of microtubules, and some are just going to keep going out, but some are going to latch on, some are going to latch on, they're going to go, and they're all going to start to find these kinetochores all over the place. And so eventually every chromosome is going to have a kinetochore on either side of this X shape of the sister chromatids. So outside every sister chromatid, eventually this prometaphase part, everybody gets connected, if you will. And so what you're going to have is this mixture of kinetochore microtubules. So these guys that actually connect to a kinetochore, these are called kinetochore microtubules. And these guys that don't connect to any kinetochore, they're called non-kinetochore. And you're like, why do we have those guys? Those guys, the non-kinetochore microtubules, actually play a very important part. They're going to help stretch the cell out, because eventually we're going to be pulling the cell into two new cells. So let's write out what we got here. So the first thing that's going on is that the nuclear envelope breaks down. And now the centromeres have migrated to the poles, and now the microtubules have access to the kinetochore region on every chromosome. So the microtubules bind to the kinetochores, which make them kinetochore microtubules, kinetochores on the sister chromatids. And then lastly, the non-kinetochore microtubules are going to help lengthen the cell as well. So non-kinetochore help lengthen the cell. It's kind of stretching it out, if you will. Okay, so prophase. We're beginning to condense the DNA, the chromatin. It's completely condensed. It's getting pretty much completely condensed by this phase. Nuclear envelope is breaking down by the time we get to prometaphase. The centrosomes that were copied, and they're growing these microtubule fibers, are now able to get to the chromosomes, the sister chromatids, and latch onto their kinetochore region, and those guys are called kinetochore microtubules. The centrosomes are additionally making microtubules that don't attach to the kinetochore, and those are called non-kinetochore microtubules, and those are helped just kind of to stretch the cell out. They're kind of pushing the cell out, which is great. So we're kind of starting to lengthen the cell a bit. All right, come back. We're going to continue more phases in mitosis. We got metaphase, anaphase, telophase, and cytokinesis. So we got 4 more phases to go, and then we will have 2 new cells. So come back. I'll see you all soon. Thanks.